Okay, I think we're set up. Ambuja, can you hear us? Okay.
Yes, now I can hear you. Fantastic.
Perfect. Perfect. Okay, so we're gonna get started with today's C3 chat. Very excited for this session because it's about research. I myself have a PhD and a master's in cancer research. But I'm not the speaker here. I'm just moderating what I think will be a fascinating discussion and really informative. So just before we get into that, this meeting will be recorded. You don't need to turn on your mics or your videos. But when we get to question and answers, feel free to turn on your mics and ask your question that way. So before we get into the actual meat of the meeting, I'm just going to share some announcements. So we are the C3. The C3 is a Canadian Cholangiocarcinoma Collaborative. We're a grant-funded research network funded by the Canadian Cancer Society and the Canadian Institute of Health Research. We won an award called the 2023 Breakthrough Team Grant, and the purpose of this grant was to build a national network to drive research discoveries and expand access to immunotherapies for people with biliary tract cancers. So since 2023, we've been working really hard in expanding our network, growing our research, and doing webinars like these to increase awareness for cholangiocarcinoma, biliary tract cancers, and also research and the importance of it. And so just some announcements before we get into it again. So this February, and every February, is actually Cholangiocarcinoma Awareness Month. There's always a ton of things that are going on, all in support of cholangiocarcinoma awareness, and I really encourage you to participate and get involved, to share some of the things that are happening through social media or with your friends and family, because really we're all here just to increase awareness for this rare cancer. And so there is a QR code on the screen, and that links to Canada's Cholangiocarcinoma Awareness Month website, and it's being hosted by the CHCC. But on that website, you'll find you'll find this map, and this map is actually an interactive map. You'll see where all of the different landmarks across Canada will be lit green in support for cholangiocarcinoma. And if you click onto the light bulb, you'll actually see if there's a local meetup planned or an event happening at that site. So definitely take a look at the website there. There's also a list of the webinars and different events and a little bit more about cholangiocarcinoma, so you could share it with anyone that's interested in learning more. This is just some pictures from last year's Light It Green event. And yeah, had a great turnout at different landmarks, and we hope that this year is even bigger. So we have some upcoming webinars with a focus on cholangiocarcinoma in Canada. And both of these are actually on that event page, or you can find it on our website, www.cholangio.ca. And so the first one is on February 17th, and it features Corey, patient living with cholangiocarcinoma from Hamilton. Also Dr. Rebecca Auer, our co-founder, Dr. Arndt Vogel, a medical oncologist from Princess Margaret, and also Dr. Robert Grant, who's also a medical oncologist from Princess Margaret. This one's going to be focused on advancing care in Canada. It'll have an update on clinical trials, on the molecular testing advocacy that we're planning to do, and also we'll hear from Corey and her life experience and going through the diagnosis. The second webinar is in collaboration with 3 different groups. So the Canadian Association for Psychosocial Oncology, or CAPO, the Canadian Organization for Rare Disorders, or CORD, and the C3. So I'll actually be presenting at this one, and it's all about talking about advocacy and how we can go from approaching it more from like a systemic advocacy point of view. We'll also have a patient advocate share her experience with advocating for this rare cancer, and it's going to be a really good discussion, I think. So encourage you to join both of those if you can. They're both webinars, they're both free, and yeah, you can just register by the link there. Okay, so those are the announcements, and I just want to introduce the speakers now for today. So we have Dr. Zaid Taha, and he is a postdoctoral fellow at the Ottawa Hospital Research Institute. He's in the lab of Dr. Carolina Ilko, who's one of the C3 co-founders as well. But his research focuses on immunology and cancer, using oncolytic viruses, or viruses that kill cancer cells. He's also looking at tumor-infiltrating lymphocyte therapy. He does high throughput drug screening. But beyond his very impressive research, he's also doing a lot in the patient engagement and education side of things. And so he's involved in many research initiatives and patient groups. He also leads the student association at the OHRI, or at least was a leader for it. So he's going to give our first talk for the first half, and then very pleased to introduce Ambush, who I think many of you know, but he's been a large voice in the cholangiocarcinoma awareness space, and he's a caregiver advocate, former caregiver to his father who passed away from cholangiocarcinoma. But since that experience, I think that he's really, used his skills and his personal abilities to promoting just awareness, research, and helping others that are going through the diagnosis as well. So really excited to hear from both of them. And I will let Zaid please take over the screen sharing. Yeah, so while you do that, I'll also mention that there's a question and answer tab on the top of the chat. There's also the chat box. So when we get to any question period, feel free to use either of those. I'll monitor that. Yeah. So I'll let Zaid take it away. Thanks.
Perfect. Well, hello, everybody. Thank you very much, everybody, for having me here. Can you hear me okay? And can you see the slide okay? Okay, excellent. So thank you, Lana, for the intro. My name is Zaid. Like he said, I'm a postdoctoral fellow here at the Research Institute in Ottawa. What that means is that I don't see a lot of sunlight. I do a lot of research in the lab, but it's okay because our days are going to get longer soon with the summer. So I'm really excited to talk to you a little bit about what research looks like from inside the lab. So how does an idea come from or go from being simply that an idea to potentially something that can change lives in the clinic? And so the slides will move. Oh no, sorry, one sec. Okay, so at this point, I mean, I'm not really sure how interactive I can be because I don't think people can turn on their mics, or I'm not sure how that works. But at this point, I like to ask what usually comes to mind when I say cancer research. And so I've kind of just like typed up and put in kind of what I've gotten in the past. A lot of people from the audiences have shouted out the unknown. When I say cancer research, some people think about difficulty, they think about communication. A lot of people shout out statistics or numbers, things like that, trials. A lot of people say care. So a lot of people are kind of thinking toward maybe the more positives, care, teamwork. A lot of people say the word journey, and we hear that a lot, journey. And I really love hope. A lot of people shout out hope. And of course, there's one more, which is human. Because a lot of people say statistics, a lot of people say numbers, a lot of people say trials, but at its core, cancer research is a human science. And as you can see, there's a lot of different things that the term cancer research can bring to mind, which is no surprise because cancer is a very complicated field that refers to over 200 different diseases. And so the endeavor of trying to understand it and treat it is just as complicated, if not more. And so when we're looking at the spheres of cancer research, of course, cancer research has a lot of pure science, but there's a lot of it that extends beyond, that pure. Sorry, a little technical difficulty on my screen. Okay, but beyond the pure science, there's a lot more. There's a very human aspect to it as well. I'm going to get into that in a little bit. But the spheres of cancer research really attempt to answer these four big questions, including but not limited to, of course. So these include: how does cancer develop? How can we detect it? How can we treat it? And then how can we help patients cope? So how can we help patients cope with a diagnosis? How can we help patients cope with their care journey? How can we help patients cope beyond their cancer journey. And so there's a lot of pure science and a lot that also extends beyond it. And so how does cancer research actually happen? So you can appreciate from here that it's pretty much a continuum where a concept is conceived, it's refined, and then through a series of steps, it's eventually brought to the clinic. So the first of these steps, and you can kind of see there's a little bit of dichotomy here. Part of it is in the lab and part of it is in the clinic. The very first of these, lab-based concepts is the sphere of basic cancer research. So I say the word basic, but it's just the terminology here. Basic, there's nothing basic about basic cancer research. It is actually incredibly complex. And in this sphere, we are trying to answer the questions: how does cancer form? Why does it form? What does cancer look like when it forms? What characteristics do different cancers have? So it's a lot of these very intricate questions about the very basic biology of cancer. And so that's basic cancer research. It's a lot of pure science in the lab. Then there's kind of this hybrid field of translational research. So we take a lot of what we learn in basic cancer research, and we try to see how we can use it to help patients. So now, based off of what we found out in basic cancer research, we start thinking, based on these observations, can we develop new therapies to treat cancer? Can we develop new ways to detect cancer? Can we link what we see in the lab to what happens in the clinic? So we're kind of moving in that direction, moving along the continuum. And last, but certainly not least, is this element of the clinical setting. So clinical cancer research, which is a lot of what people have a lot of questions about. So a lot of the questions here include, will our new therapy benefit patients? How are different groups of people able to access or afford different treatments? So it's not just a patient-centered approach, it's a people-centered approach. It's a population study. There's a lot of economic studies that go into it too, because cancer care is not very simple. There's a lot that goes into cancer care. So we try to look at every element of it as well. As well, we look at some sort of epidemiological elements here as well. So how do different behaviors affect cancer risk and so forth? So the spheres of cancer research are very, it's a research continuum. And cancer care itself is a continuum, and a lot of it starts with research and ends at clinical care. And I do want to emphasize, of course, that we have to have the utmost respect and appreciation for our physicians, our oncologists, our nurses, our care team, the administrative staff, because they do the life-saving, right? They do a lot of that life-saving business. But we should also not forget and never diminish the fact that everything that's used in the clinic came from an observation that was made by or tested by or validated by a scientist. And so in that regard, I do also want to kind of emphasize that cancer research is cancer care. And that's what I'm hoping this slide can help to kind of convince you about. So we're looking at here are the mortality rates or cancer-related mortality rates from the Canadian Cancer Society Statistics Report in 2025. And the top is females and the bottom is males. And it goes as early as 1980 or 1984 all the way up to 2023. And as you can see, there is a nice decline because a lot of what we're doing in the clinic is making a big difference. So survival rates, outcomes, everything is a lot better. And I want to show you how, because of science, because of research, this is why we see, these declines. So one of the easiest examples I want to give you is that in the early 90s, the anti-smoking campaigns became a real thing because after, decades of research, we finally realized that it's not really too cool and it's also really bad for your health. And so with those massive anti-smoking campaigns, overall outcomes in cancer care were a lot better. Treatments became better. People were able to tolerate treatments better. The incidence or the occurrence of a lot of different cancers in the lung, in the throat, in the mouth decreased significantly and became more manageable. And this is just cancer care, right? So we're not even looking at the excellent outcomes that we saw in other fields like heart health, for example, cardiovascular health. So that's one example. Now, we also realize early on that if we improve our screening for cancer, we can detect cancer sooner and we can treat it better, right? Because earlier detection means much better outcomes. We also learned that now we can have new combinations of different treatments together, which also lead to significantly improved outcomes, as well as better safety profiles. So patients not only would have better outcomes, but they would respond and have better side effect profiles. So they would tolerate their treatments a lot better. It was a lot safer for them. Now, this is a bit of a case study here, and I really want to emphasize it because I kind of invoke it a little bit later on. So in the very early 2000s and even in the late 90s, this whole idea that if the immune system is activated, we can get better outcomes in cancer care was a documented thing. And then one of the most landmark publications came out in 2001, and it published this concept of PD-1, PD-L1. Now, we don't have to get into the specifics of what this is, but this is basically a drug that activates the immune system, and it helps the immune system, when it's activated, destroy cancer cells. And this was published in 2001. So that was just a concept in 2001. In under 10 years, it was about 6 years later that the first clinical trial happened. And since then, there have been thousands of these clinical trials. And it very rapidly, in under 10 years from that point, it received clinical approval. And this wasn't just one clinical approval. It was 9 clinical approvals across 9 major cancer types at the time. And as of right now, this concept, this PD-1, PD-L1 therapy, which activates the immune system, is not just clinically approved, it's actually a standard of care. So it's not just something that patients can be offered, it's something that they are almost immediately treated with because of how effective it was. And just seeing in under 2 decades, we went from a concept to standard of care. So that really is mind-blowing how fast we can go. And you can really see how that kind of decline in mortality and the improvements in outcomes and survival are really a result of cancer research. Now, since then, immunotherapy became a really big thing where we of course understood that the immune system plays a huge role in the treatment of cancer as well as other things. And so a lot of different immune activating things became a very important part of preventing and treating cancer. So this isn't necessarily a cancer treatment, but it was a massive preventative campaign for several different cancer types. So Gardasil, which is for the human papillomavirus or HPV, it was originally approved in the very early 2000s just for little girls to prevent cervical cancer. But we quickly saw that, if we approve this for boys and girls and even adults, we can actually significantly improve outcomes for almost 50 different cancer types. And that was also part of this downward journey that we're seeing. And as of the 2020s, we have these almost science fiction therapies that are they're moving mountains in the clinic. And so we're living in a time where things are moving very fast with these brand new therapies that are being made increasingly more accessible and available to patients. And I think this has really brought about the advent of precision medicine. And so what is precision medicine? So precision medicine is basically where we try to tailor the treatments to the patient. So this is not just, if a patient has this specific disease, we give them this treatment. No, now we're looking in the genetics of the patients. We're looking in their molecular profiles. We're looking in their environment. We're really dissecting that disease and why it happened. And that is how we try to treat it. We treat it at its core. It's almost like a surgical strike on that tumor. And precision medicine very much moves away from the one-size-fits-all model of trying to treat disease. And we're seeing this increasingly in, of course, cancer research, as well as a lot of different other diseases. And I want to give you a really exciting case study, and rather a success story of the power of precision medicine. So this was a paper that was published in 2022. And at the time, it created ripples in the scientific community. So what happened here was we had 12 patients who had an advanced cancer, and it was cancer in the gastrointestinal tract. And these patients were set to come in, and the treatment plan was 6 months of this drug called dostarlimab. It's an immune activating drug. And then the idea was after 6 months of treatment, then the patients would have to come in and receive follow-up radiation, surgery, additional care, because the expectation was that they would need this additional care. And so what ended up happening was these patients with the advanced cancer came in, they were put on 6 months of dostarlimab, and then they came in for their follow-up so they could get scanned and then, follow up with radiation, surgery, additional care. But what was really mind-blowing was that not a single one of these patients had any trace of cancer in their body at that time. So it was a 100% complete response. Everybody had gone into remission. And I think this really emphasizes just how powerful precision medicine really is. So at the time, a lot of the scientific community was, a lot of people critique the study for, saying things like you cherry-picked patients to make your drug look like it worked better. But I argue that is, not the case at all. The idea is we want to pick we want our drugs to have the best chance of success in patients who would most likely respond, because we want people to benefit. And that is precision medicine, is where you pick the treatments specifically for the patient. You can't just assume that anything is going to work for everybody. And so I thought that was really exciting and just a really interesting example of how precision medicine can really be a success. So how did they do it? How did they really get to this point? How did that concept end up being a life-saving therapy, or an almost a 100% response in all of these patients? So we go through this method in research called the scientific method. It's a very systematic way of coming up with an idea and testing it, and then refining our idea and testing it again until we have this perfect recipe of something that works for whatever it is, whether it's cancer research or whether we're studying something about gravity and physics or whatever field we're in. The scientific method is a very standardized process. So in cancer research, how this comes about is, first and foremost, we have to observe something that makes us come up with a hypothesis. And a hypothesis is basically a prediction. So let's say we observe that in a lot of individuals that have a suppressed immune system or a weak immune system, there are higher rates of cancer. And in a lot of individuals that have a very active immune system, there are lower rates of cancer. So then our hypothesis can be the immune system can kill cancer cells. And so we can very easily test this in the lab in very, very simple things, like just putting immune cells with cancer cells in a little dish, and then we can see if the immune cells can kill the cancer cells or not. And we can eventually validate and refine this hypothesis. And this just means that we kind of clean up our idea. We talk to our colleagues about it. We fix it up. We test it out a little bit better until we have a more definitive concept. So instead of saying, okay, immune system can just kill cancer cells, let's refine this better so we can try to make something that can actually benefit patients. So I have found an immune activating drug, and I think this immune activating drug can help my immune cells kill cancer cells. And so we test this out some more. We can do it in more elaborate models. So for example, in animal models of disease, so for example, in mice that have cancer, if we give them an immune activating drug, will this help them? And so forth. And let's say this goes according to plan, and we now test it out in more mouse models, for example, or other different animal models of disease, multiple different cancer types. Not only are we interested in, does this drug work? Does it actually help treat cancer? But how safe is it? How are these animals tolerating this drug? How is the drug actually working? So now we can start looking at mechanisms. So we're just refining the idea. First, we have to prove that it works. Then we try to really understand how it works. And if we can determine all of these things and also definitively say it's safe, now we can approach Health Canada, for example, and say, hey, we have this awesome concept. It's safe. It's fantastic. We really believe it can help patients. So we want to put this through into the clinic. And so now we can move into the clinical phase of research. So there's a lot of different phases of clinical trials. Clinical trials are basically how we test out whether or not our therapeutic can benefit patients. The very first three phases, phase 0, 1, and 2, basically are centered around safety. There is nothing more important than patient safety in the entire course of a drug or a therapy. So we must make sure that the drug is safe in humans. So we would test this drug out in, for example, patients, and see how well they tolerate it. We are hoping that everybody would be happy and safe and they can tolerate the drug. And of course, if we see that there's any benefits on disease progression, let's say that this drug is actually reducing disease progression or leading to regression of the tumor, for example, that's also fantastic to know. But first and foremost, safety. Finally, we can test this out in the phase 3 trial, which will directly compare our new treatments against the standard of care. And ultimately, we aim to show that our new treatment is not worse than, or better than at least, the standard of care. And eventually, if we can prove that, then we would have to approach these government agencies to try to get approval for clinical application of our drug. Now, of course, you can appreciate that this process has a lot of steps, and of course, it is incredibly time intensive. So the actual basic and preclinical research will take anywhere from 1 to 5 years, usually towards the upper end, usually at least about 5 years. And then if we do get something into the clinical research phase, to actually get it to approval, if it actually gets there, it will usually take no less than 5 years and oftentimes more than 10 years. So it is a very time intensive process. What's more is that really high quality research takes a lot of time, but it also takes a lot of money. Research is no cheap endeavor. Cancer research itself is no cheap endeavor because a lot of our models, a lot of our a lot of our reagents, everything is very, very specialized. So this is just an example of what a budget looks like for the purposes of this presentation only, but it is based on values that are relatively representative of how much it costs, for example, to get some of these things. So, for example, to run an academic lab, a research lab that comes up with a concept and tries to test it out before we get to the clinic, it could take anywhere from about $200,000 to $350,000 a year just for 1 year. And this is for very basic things like to have students and to just have the basic equipment and ingredients or reagents to do the science that we want to do. So it's absolutely no cheap endeavor. And now if we're moving towards the clinic, you can appreciate that this is a clinical trial cost per patient per year for something as intricate as what the C3, for example, is planning. So for a more advanced clinical trial with one of these innovative biological therapies, but still these new innovative biological therapies tend to be the ones that are very, very effective. And they're very expensive naturally to do. So it's important to appreciate just how much it can take to make this happen. And because we don't necessarily have, let's say, like if you don't have an industrial partner, like a company that is sponsoring these trials, an academic institution or a university, for example, or a hospital needs to really be careful with how they spend their money. And so it's really important to appreciate just how much some of these things can cost, easily upwards of $1 million a year if you even want to tread into the field of clinical research. And so it's really important, I think, the real takeaway from this is, no matter the cost, of course, people will keep working and try their hardest and try to make things a reality. But it's, of course, very important to advocate for research and development as much as we can, because funding cuts effectively tear down our research and they tear down our future. So it's important to keep that in mind. So talking a little bit more about some of the science that I actually do and some of the stuff that I work with in the lab. So a little bit away from how does cancer research happen, more about a show and tell of some of my work. So I work a lot on what we call immunotherapy. And as the name suggests, of course, it's a type of therapy that involves the immune system. So what is the immune system, right? So it's a very complicated thing. It has millions of different elements, millions of different moving pieces constantly working together to essentially protect our body from internal and external invaders. But at its core, the immune system is this system, this very powerful system that prevents invaders from taking over our body, whether they are bacteria, viruses, parasites, or even internal things like cancer. And there's several different moving parts of the immune system. And so our defense team lineup has these like major types of cells. I just like talking about this. I'll go through it very quickly. But we have these things called natural killer cells or NK cells. And as the name suggests, they're very effective at killing things that don't look right in the body. Infected cells, cancer cells, and they deliver this figurative kiss of death. And they deliver these very powerful biological substances that can kill abnormal looking cells. We have these macrophages. So they're these big, hungry guard cells that monitor everywhere in the skin, in your tonsils, in your eyes. They're absolutely everywhere in the body, and they eat up things that look abnormal, and they destroy them. We have these dendritic cells, and these things are a work of art. They're some of my favorite things to work with. I work with them all the time. They have these long-looking tentacle extrusions from their body. What they do with these is they kind of grab things that look abnormal and they rip them apart and they kind of wave their arms around, and they alert the immune system that, hey, like what we have here, this is abnormal. You have to look out for this and you have to destroy this. So dendritic cells play a really big role in activating the immune system. Now we have these humble yet incredibly powerful little cells called T cells or T lymphocytes. You must have heard about this, or you might have heard about this rather. They're dormant cells, but once active, they're almost unstoppable. They can take care and adapt to almost any situation. They're responsible for immunity that you get from vaccinations. They're responsible for killing cancer. They're responsible for killing infected cells, from fighting off bacteria and viruses. They can do everything under the sun. And B cells are their counterparts. Instead of directly attacking and destroying things, they secrete these little things like homing missiles that we call antibodies to find and destroy their targets. And so the immune system is very, very well equipped to destroy things that shouldn't be where they are, right? And so with this powerful defense team, the immune system is always watching, right? There's this concept of immune surveillance that has become increasingly more obvious to us since the 60s, since the very, very first kind of paper was published on it. The immune system is always watching. It's watching everything, everywhere, all at once. Why? Well, because we have hundreds of trillions of cells in our bodies. One of them can easily get infected. We might easily introduce something that is like a microbe or a pathogen into our body. One of these hundreds of trillions of cells themselves might actually do something wrong and will need to be corrected. So the immune system is always looking out for us. And so in the concept of in the sphere of cancer, let's say, let's look at this analogy, this very simple analogy. If you have this cell, this normal cell in the body, and it's dividing into these other normal cells, but oh, something happened, and for one reason or the other, one of these cells is abnormal. Now, we don't know what it is. We don't know if it's a cancer cell. We don't know what happened, but for one reason or the other, one of these cells is not right. And so the immune system would come in and it would detect it and it would effectively destroy it. And the immune system is exceptionally powerful at being able to do things like that. And it's especially, I mean, I say this because I'm biased to T cells, but I think T cells are outstanding little organisms that just do, one second, I'm gonna try to play this video for you guys. Yeah, T cells are just absolutely capable of destroying tumors. And so this is a video that I pulled off of, at the time it was Twitter, it's now X, from the Ritter lab in this, I think it was in the States. And what you're looking at here in blue are cancer cells, and these pink circles or pink spots are T cells, killer T cells. And you can see here, yeah, sorry.
Sorry to interrupt. We don't see the video, at least I don't see it on my screen.
Okay, no problem.
Maybe
well,
if you stop sharing and then share your screen instead of the presentation, that could work.
Okay, let me try that. Sorry, everybody, just give me one moment. Okay, so if I go like this, can you guys see it like that?
Yeah, much better.
Okay, perfect, excellent. So these pink spots are T cells, killer T cells, and these green big blobs are cancer cells. And when these pink T cells attach to the cancer cells and they detect them, you can see this like red wave of almost death that they send into that cancer cell, and they absolutely destroy it. And that was one or two T cells working over there. And on the right-hand side, you can appreciate more T cells doing that same job. And the cool thing is that T cells can go around doing this to hundreds of or thousands of cancer cells at a time. They don't just do it to one and stop. And so the immune system is very well equipped to deal with things like this. But the reality is that one of the hallmarks of cancer is that it can avoid the immune system. It's called immune evasion. And it takes on sometimes a cloak for whatever reason. And so again, in this analogy, you have the cell that's replicating, and one of these cancer cells is abnormal, but for one reason or the other, it's acquired a cloak or camouflage, and now it's not very detectable. And so the immune system can't really see those camouflage cells. This is what the immune system sees. And if the immune system can't see it, it can't kill it, despite being very well equipped to destroy. And if this is the reality and the immune system can't deal with it, unfortunately, that is one of the ways that cancer can establish itself. And this is why immunotherapy is such an incredible field and such a powerful field, because it's not just utilizing the immune system to kill cancer cells, because that's what it already can do, right? We're not telling it to do its job. We're actually trying to find ways to stimulate it, to be able to see and recognize those hidden tumors and to destroy them. And that is the power of immunotherapy. And what I think is exceptionally exciting is that Ottawa is a hub of immunotherapy expertise. And that is why I am so lucky to be where I am, because not only do I have my incredible mentors to help me, but we have everybody and all the equipment here to make our research a reality. And so the Ottawa Hospital Research Institute has about 59 research labs. It's the fourth in Canada for research funding. And a big part of that is cancer research. And because of that funding, we have made such huge economic impacts in Ottawa. And we have over 800 active clinical trials, many of which are also cancer focused. And so it is a very, very active research institute that is very impactful. And what's most exciting is because we're located in a hospital, we're able to quite literally take ideas from bench to bedside. So we can come up with an idea, we can translate it, we can prove that it works. And then we even have a biotherapeutics manufacturing center that can actually build and manufacture our own therapies right here in-house, so we can give them to our patients in clinical trials right here. So we really can translate science from the lab to the clinic. We have done this in the past, and we are continuing to do this now. So I'm going to talk in the next 3 to 4 minutes just about a little bit of the science that I actually do. And so I decided to talk about TILs or TIL therapy because a lot of you might be familiar with this. It's one of my big areas of focus, and the C3 is very focused on TIL therapy. So TILs, as the name suggests, are lymphocytes, which is another name for specialized immune cells that have infiltrated into tumors. So the immune system will detect and try to kill tumor cells, but sometimes these immune cells, when they get into the tumor, they can get overwhelmed and suppressed. And so you have these tumors that have a bunch of immune cells inside that are kind of dormant and inactive. And these immune cells also happen to have these very active, very anti-tumor capable T cells. And so what TIL therapy aims to do is we try to use these as a living therapy. So the TIL therapy process starts from when a patient undergoes surgery. So when surgery is happening and the tumor is removed, typically that tumor can just go for diagnostics and then it's destroyed or discarded. But we take that tumor material, we process it, and then we grow out those immune cells from the tumor. We grow out those TILs, and we grow them to extraordinary numbers. So we have hundreds of billions of cells now that are fundamentally anti-tumor in nature because that's where they were. And we grow them up in these bioreactors, and then we end up infusing them back into the patients. And now these cells came from tumors, so they know what the tumor looks like. They know where the tumor is. They know what they know everything about the tumor. And now when we're giving them in these extraordinary numbers, they can home in on these tumors and destroy them. And they have been very effective at doing that. And it's a very, very exciting new wave of therapies that are beginning. And what's really exciting is that the C3 was actually awarded a really big sum of money so we can make this a reality for patients right here in Canada. And my work specifically focuses on how I can make the strategy more successful and also more accessible to Canadians. The other thing I'll touch on, and this is the last thing before I hand off to the next speaker, is oncolytic viruses, so cancer-killing viruses. So if anybody here has had either the monkeypox or the smallpox vaccine, you have received this virus that you're looking at on the screen. So this is vaccinia virus. It is a very safe, very big virus that was essentially the reason that we eradicated smallpox. So it's exceptionally safe, and we owe it a lot for saving our lives. But because it's so big and so safe and so versatile, we were actually able to engineer it so that it can become a cancer-killing virus or an oncolytic virus. And what that means is that if you take a normal cell and a cancer cell and you infect them with this new engineered version of this virus, only the cancer cell will be infected and will actually end up dying, but the normal cell will be completely unharmed. And this type of therapy is really exciting because we're taking what nature has already provided, these very, very intricate almost hijackers of cells, these viruses. And we've simply engineered them so that they are safe for us, but bad for cancer. And we're kind of repurposing what already exists. And this has become a very exciting field, and it's very active. And there's thousands of clinical trials out there that show how effective this kind of treatment is. And so I'm actively working with my two supervisors, Dr. John Bell and Dr. Carolina Ilko, into making some of these viruses into cancer-killing battleships. So not only will these viruses go and rupture these cancer cells, they can also alert the immune system to tell it that there is a cancer here. These viruses can also produce anti-cancer biological substances in the tumor. So instead of, for example, giving a drug intravenously to a patient and having to deal with those side effects, now that is being produced by the virus in the tumor. Also, because these viruses can destroy blood vessels within the tumor, they can starve and suffocate cancer cells, ultimately destroying the tumor through all these different mechanisms. So it's a very up and coming field, and there's so many different things we can do with this. And I mean, I'm obsessed with these oncolytic viruses, and I'm actively working on them. So happy to answer questions later. But a quick trigger warning on the next slide, just to show you what the impact of this can look like. So in one of the clinical trials where this specific vaccinia virus was administered to patients who had liver cancer, not only did that patient's liver cancer regress, so it was shrinking, but the distant sites of where that tumor had gone, so the metastatic lesions like in like near the neck of this patient, it had also completely gone away. So these oncolytic viruses do have a lot of therapeutic potential. And so, of course, science is fantastic. There's a lot of skills we learn at the bench, but our team is very excited about what we can also do beyond the lab bench. So we love engaging with our community. We love engaging with these different agencies so we can talk about our science, so we can also spread awareness about our research and talk about how important it is to keep research and development alive and funded. Knowledge exchange initiatives, we have a lot of these things where we like to talk about our science to the community. I've highlighted the Café Scientifique because this is coming up, and I'm going to hopefully have Leonard circulate the link to everybody in the C3 as well. This is going to happen in Ottawa, March 7th. If anybody, if you have ever had the question, what does cancer research really look like? What does the equipment look like? Can I look at these cells? What do these cells look like? Then this is the right event for you. We have this interactive lab tour, and it's just a really exciting thing. And again, happy to talk about what we do beyond the lab bench as well, but I just want to emphasize that we are not only excited about working together with other scientists, but most importantly, we're excited about working together with our community so that together we can co-create the future of cancer research. And with that, I want to thank you for your attention. My email's there if you have any questions. Happy to answer now or shoot me an email. And yeah, thank you for having me here.
Thanks so much, Zaid. I think we're going to save the questions to the end, and then maybe it'll be sort of like a panel. People can ask either Ambuj or Zaid their questions. So yeah, without further ado, I'll let Ambuj unmute himself. If you can just introduce yourself again, just for to refresh our memories.
Sure.
But this next talk is going to be coming and looking at research from a different perspective, caregiver's perspective. So let me just spotlight you, and I'll let you take it away. Thanks.
Thanks, Leonard. And I actually have a couple of slides I wanted to share. Could you add me as one of the presenters so I can share my screen?
Okay, while I do that,
if possible. Yeah, I mean, I can start. That's fine.
yeah.
So thank you very much, Dr. Taha. That was absolutely incredible to see all the great work that's happening with the TIL and the oncolytic virus. The latter being the more interesting one as well. It seems like it's very new and upcoming. So thank you very much, Leonard, and to C3 for inviting me for this. And so my background is in engineering, and I work as a senior technology leader in a financial institution. But in 2019, our life as a family shifted and changed when my father was diagnosed with cholangiocarcinoma, and he passed away in 2022. And that experience really changed how I think about healthcare and cancer research and what it truly means to drive meaningful impact across the cancer journey for families, patients and families alike. So I became involved as a mentee with the Cholangiocarcinoma Foundation out in the US. I had a mentor assigned to me, and over time that evolved into me becoming a patient and research advocate as we were looking for specific solutions for my father. And in fact, I have that foundation to thank me for helping me find my voice. And I think Leonard would like to say something. He would like to voice something. Oh, you've given me.
Yeah, you should have access to share your slide now.
Fantastic. Thank you. Thank you very much. Yeah, so I found my voice when I had the opportunity to lead and co-host the foundation's first Canadian chapter of the patient support group back in 2023. And since then, I have served as a board member and a patient advocate across different nonprofits, from local to international, so from Ontario, Alberta to US and India. But what I was looking for was something that was more closer to home, something that was more local, where I could contribute more deeply to the research ecosystem here in Ontario, because that is what benefited us and my father. And that's where my work with the Ontario Institute of Cancer Research, based out in Toronto, began. As I became more involved within the patient advocacy space, I realized that the caregiver perspective, especially in rare cancers, was often missing. So that gap is what led me to become involved with the PFAC, which is the Patient and Family Advisory Council at the OICR, at the Cancer Research Institute. But this was not specific to just cholangiocarcinoma, but across any type of cancer research. So I really want to build on what Dr. Taha was saying on what he's shared from the research side. as to why, how do we take our intention to real world impact, but from a different perspective, not from a scientific perspective, but from a patient and caregiver perspective. So I'd like to break this down into three different categories. So the first one is building relationships. So one of the things that I realized is research becomes a lot stronger when the relationships can replace the distance. So one of the most powerful examples for me was when I was with OICR, is they have a lab that's led by Valentina and Dr. Kushku and their incredible team, and that's specific to cholangiocarcinoma, which obviously is very near and dear to my heart. And they were doing excellent and important work for advancing research in cholangiocarcinoma. So as a patient partner with the OICR, I was working with them quite closely at times. And it's worth highlighting that the C3 is the one that's supporting the funding for this research as well. So it came full circle for me. And, we were talking about building relationships. Last year, C3 hosted a conference, and through that partnership, we had some really, really meaningful conversations happen. What stood out to me was it wasn't just about the science, but it was the intentional effort that that this group of people that were working on in this lab wanted to connect with patients and caregivers. So OICR and this lab, they organized a lab tour for the patients and caregivers that attended this conference. And for many of us, it was the first time we actually saw where the research actually happens. We always imagine that it's just someone in a white coat, a lab coat working away, but it became an abstract idea to something that we could relate to. But real people work with urgency and curiosity and care. And then the second piece was at the conference, Dr. Kushku actually gave a very deeply heartfelt presentation of what their lab was actually working on, specifically for cholangiocarcinoma. And what stood out for me was it wasn't just about the research itself, but why it mattered to her and her team, and how meaningful it was for them to see the patients and caregivers engaged in the work that they do. And I can tell you, speaking with a lot of the patients and caregivers there, everyone in that room felt the passion and the dedication that they have for this work. We felt that there was actually hope, and hope that people are doing this work, are not detached from the human aspect of it, that they genuinely care about finding answers and cures. The other piece is, about building relationships, is the patient partnership. They welcomed me to a lot of their research meetings. They made me part of their lab community, and even down to inviting me to their Christmas potluck. So that was very nice. But that openness of the researchers and scientists wanting to work closely and collaboratively with the patient community, it opened my eyes just to see how brilliant and driven these people are that are advocating for cancer research, not just professionally, but also personally. And that relationship is what is the foundation for everything that will follow. The human connection really does help strengthen the science. The second piece is the diversity of experience. That's really important. This is what the patient perspective brings into cancer research, bringing in different points of view, because you could have one diagnosis, but you will have many different journeys and solutions or realities. So the one thing that I learned, and it humbled me a lot, was that as a patient and a caregiver advocate, that my perspective wasn't just based on my own experience. I mentor caregivers as well, and working across multiple nonprofits and working with other patient community partners, I've actually had the privilege to hear dozens, if not more, stories of heartbreak, of resilience, of frustration, but yet still hopeful. So when I offer feedback or I provide feedback or insight, or have any discussions where I'm bringing in the perspective, it's not anecdotal. It's actually a culmination of lived experiences from people from different disease stages, from different socioeconomic backgrounds, geographies across the country, and even cultural context. And all of that makes a difference. And this is especially important in rare cancers, where voices are already limited. So how does this how do I bring this experience in to real world life? So at OICR, I have been able to contribute to the strategic conversations. So they have a strategic plan for the next 5 years, and the patient perspective and the caregiver perspective was brought in. And it helped identify that the organization has to look at greater awareness towards rare cancers, that there has to be ongoing equity in cancer research and treatment, and ensuring that research translates across urban and rural settings. Because as lucky as we are living in Toronto or in the heart of Ontario, the folks that are impacted by this are the ones that are most impacted by this are the ones that do not have access to the main cancer centers. So, and when I look at my peers within the PFAC and the broader patient community, I see that each person brings a unique challenge and with it, potential solutions. We have had current and former patients in the patient community that speak up, and current and former caregivers. And listening to that collective voice isn't always easy, but it is definitely essential. Because, like I said, two patients can have the exact same diagnosis, but their cancer journeys could be completely different. So it's really important that we ensure that research accounts for all of this, or else we risk becoming a very narrow focused in the research and less effective in the actual real world scenario. And then the last piece is around translating patient perspective into the actual real world impact. So the theme of today's virtual event, the webinar, is how research translates into real world impact. And my perspective on this is to show a couple of real life examples of actual output of how that actually helps. So at OICR, we're beginning to see a lot of patient perspectives shape how research is done at the Cancer Research Agency, how research are prioritized, how questions are framed around the cancer research, and how the feasibility and impact are assessed for these treatment options. Now, I'll give you a couple of examples as well. So in 2023, I believe it was 2023, the patients and caregivers were actually invited to contribute their personal cancer stories as part of a funding proposal for C3. Leonard, I don't know if you recall, but this was a great opportunity that we were able to bring in our perspective, our lived experience. These stories were not an afterthought. They helped articulate why their research actually mattered and whom it was going to assist and help. And that proposal was successful, and they received the grant. And outside of that, there's another example where a lot of the work that my peers and I are doing is we're reviewing research papers, medical publications, preclinical trial applications. So we have research groups that approach the myself and the PFAC, the patient community, asking for feedback. And our input has really helped improve some of the design principles, improve the consent language and clarity, add depth to the quality of life metrics, bring more realistic assumptions about the treatment burden, and surface some unrealistic protocol expectations based on real life situations that we as caregiver see our loved one go through the side effects of any drug treatment. And the last example that I'll mention here is at the government level. So bringing in the patient perspective is really important when you have cancer research agencies or institutions or nonprofit organizations vying for government funding. Like Dr. Taha mentioned earlier, that it is imperative that we have funding to support the amount of cost, like $1 million per month or per year per patient is a significant amount. So more recently, when we are engaging with the Ministry of Finance for budget approvals and strategic planning, they consistently ask for the human element, not just the science and the research, but they want to see someone within the patient community vouch for what any organization is trying to do, because it helps them understand how to make decisions. So as patient partnership matures, I believe that we are going to see even stronger connection between the patient input, the research design, and the funding success, and actual real world outcomes. There is I recently spoke with one of the original co-founders of OICR from 20 years ago, and he offered a line that's really stayed with me, and I'd like to share that. He said that the patient perspective does not slow down research. They actually need to be embedded and included from the very beginning of the cancer research lifecycle, because they help it go in the right direction. So I feel that the patient and caregiver voices can and have been making real world impact from a different perspective. It makes the research become more relevant. The trust increases between the research and scientific community and the patient community, all with the hope that we are going to have improved outcomes. So with that, I'm very open to questions. Leonard, I'll actually pass it over to you if you want to open it up for Q&A.
Yeah, for sure. And thank you again for sharing your experiences with being involved with research. And I think it's really important that we advocate and try to get more patients and caregivers involved in this, because myself being a researcher for more than 10 years, a lot of those years were spent just in the lab without any input from patients actually. And we would have learned a lot and done a lot of things differently if we did have that, essential perspective. So I will open up for any questions, and feel free to use the chat. I'll read out Heba's comment just so everyone can see, but she said that we hope that all oncologists over the country will educate to give their cholangiocarcinoma patients the proper treatment at the proper time, tailored for them specifically with timely screening, to give hope along the way. And unfortunately, oncologists that treat cholangiocarcinoma patients usually do it according to statistics and provincial resources. But yeah, I hope that we can change that too. I do have a question for Zaid. So what's one way that patients can get involved in research from your perspective, and in the non-translational phase of research?
That's a very good question. So if patients are looking to get more involved in research at not necessarily the clinical stage, but even before, we work a lot with patient partners as part of the Canadian Cancer Society. So CCS, the Canadian Cancer Society, has a lot of patient partners that sit on a lot of different grants application panels. So it's not necessarily just scholarships for students, but really big operational grants that fund labs and institutions have patient partners as part of that review process, so that there's transparency, so that you have the patient perspective, so that you can really incorporate that human element into that review process. That's one big thing. I think a really important and really exciting avenue for involvement is also attending certain conferences. So scientific conferences, specifically in cancer, like BioCancerX, which is coming up in March. They have something called the Learning Institute. I think it might be too late to register now, but for subsequent iterations, You can get matched up with like a researcher, for example, and then attend the conference and go to all these talks. And your scholar or your academic scholar partner would help to explain some of these concepts in more accessible language as well. And you just have the opportunity to see some of this research firsthand, talk with these scientists, talk with the clinicians. And yeah, I would encourage you to reach out to either the conferences or scientists like myself to help give you those resources, or even CCS. But there's increasingly more and more opportunities for patients to get involved.
A lot of the times also, like when you're admitted to a hospital, you might be given an option to participate in research studies, or like can you be contacted? So we always try to advocate that, yes, you should, if you're willing, if you're up for it, to do that, because a lot of the research that we use comes from also patient samples, and so samples from tumor biopsies, cell lines, it all helps with these studies. I do see a question in the chat from Heather and Ivan. I'm going to read the first one, and we'll just go through them in order. But is it true that TIL therapy is less effective for cold tumors like cholangiocarcinoma than hot tumors? I think, Zaid, you might be able to answer that one.
Yeah, that is a very good question, Heather and Ivan. Thank you for bringing that up. So in the context of any immune therapy, if the tumor is fundamentally colder, that's a very good use of that terminology. If the tumor is fundamentally colder, it does tend to have slightly, or it does tend to have dampened outcomes, but that's not necessarily the case, because there was a recent clinical trial. It wasn't a cholangiocarcinoma trial. It was a colorectal cancer trial. And it was done using TIL therapy for patients who had cold tumors. And those patients still saw quite a dramatic benefit. So that being said, and I think if we can kind of translate what we've learned there to cholangiocarcinoma, even though it's a different cancer subtype, I think there is still a lot of benefit for TIL therapy, even if the cholangiocarcinoma is colder. So probably less effective than if it was a very hot tumor, but there's still benefits.
Yeah, and I think that's, at least for the TIL trial that we are leading and developing in Canada, we're looking at ways to select for those immune cells that are going to do the best in cholangiocarcinoma tumor that's specific for the patient. So finding the ones that are the most active immune cells and expanding those specifically. So I think the TIL therapy is very promising, and it does represent like a not specific approach to treating tumors. And so there are also many ways to make a cold tumor hot as well. And so a lot of avenues to attack this question. The other two questions are a bit more personal, so I don't want to announce them on YouTube since we will be posting this online. But maybe I can just try to reword this third one. So it's asking if there's any research in Canada that is similar to what's going on in China, where they are giving cancer cells, they're exposing them to allergens like pollen, peanuts, and looking for an immune response. Yeah, do you know if there's any sort of research going on in Canada that's like that?
So I think I know the clinical trials that they're being referred to. It's a very interesting concept. I don't know if there's anything actively happening in Canada. There might be trials in the States, like a little, not in Canada. But interestingly, oncolytic viruses do something similar. The same way that pollen, peanuts, and certain allergens can be extremely stimulating to the immune system, viruses are a natural, huge threat to the immune system. So if the immune system sees a virus, it's going to go absolutely wild. So oncolytic virus trials, which are ongoing in the States, and there are some in Canada, are probably the closest thing to that, in my opinion. But I need to read up to understand a little bit more.
Okay, great. I do have one last question, maybe for Ambu, since we're going over time, but you don't have any scientific research experience per se, correct? So how does your personal, lived experiences and also just professional abilities, how would you say that contributes to being a patient partner on different research teams? And also, what would you say to someone that might use that as an excuse for not joining?
That's a great question, Leonard. Yes. So a lot of times when you're a caregiver or even a patient going through the cancer journey, you end up picking up a lot of lingo that makes you sound like as if you are in the healthcare field, because you're talking with doctors, nurses, oncologists, all the time. So I picked up a lot of the medical lingo and jargon from that. And because also I was a research advocate, I was in a lot of trainings as well. What is cancer, cancer 101, and what are clinical trials? So I took the opportunity of being in that position of educating myself uh, so that I could at least understand at a basic level. Now, how do I actually translate that to the other areas of my advocacy? Well, so I would say that we have a lot of technical skills that we gain in our professions, and then we also have soft skills. And soft skills are transferable. So if you have the opportunity of speaking or doing organization or project management or marketing or sales, whatever it may be, everything that you have done in your life up until now will have an impact, a positive impact on how it can support you in whatever you would like to do as part of patient advocacy. You do not have to be in the medical field. And I can tell you from my personal experience, working alongside and volunteering alongside a lot of the patient community, majority of them do not have a medical background, yet they find what has always worked best for them and what they're good at to apply it to research advocacy and patient advocacy.
Yeah, I totally agree. And I think that as researchers and scientists, sometimes we even get a little bit narrow-minded or too focused on, we're not broad thinking. And so really having that diverse skill set of opinions and experiences from different fields and sectors really does help make research more full and give the full picture. So yeah, with that, I'll also say that the C3, we often get research proposals or different investigators that reach out to us looking for patient partners or people to be involved. And on a couple of occasions, I've sent some names to researchers and they've been involved in the grant process. So if you are ever interested in getting involved and joining a research team from the patient side of point of view, please reach out to the C3. I'd be more than happy to walk you through the process and try to pair you up with a researcher. Okay, so thank you all for staying a little bit over time. Thank you, our guest speakers, Dr. Taha and Ambuj. And yeah, we'll see you at the next C3 chat.
Thank you very much.
Bye, everybody.
Bye.
Thank you so much. Bye.